This video for Gen Bio at RCBC will discuss differences between prokaryotes and eukaryotes. Now, there are many more differences that I won't discuss here, but these are a few that I do want you to know. If we take a look at this prokaryotic cell versus a eukaryotic cell, one thing we see immediately is that there is a nucleus here that doesn't exist here. That allows transcription and translation in prokaryotes to happen at the same time. It's not really well illustrated here, but transcription and translation, as we start to make the mRNA strand, the ribosome can jump right on it before we've even finished the mRNA strand. So it can happen at the same time and, of course, in the same place. Whereas in eukaryotes, transcription happens in a different place than translation, so of course it can happen at the same time. Another difference is in the promoter region and how the RNA polymerase finds the promoter region. So the promoter regions are slightly different where they are, and if the gene begins to be transcribed here, a eukaryotic promoter region might be here. Maybe there's another region here that helps with the promoter, which helps with RNA polymerase, and maybe it's so many base pairs upstream in prokaryotes and a different number of base pairs upstream in eukaryotes. So the details of that don't matter. But one thing I do want to point out is that there's something called a sigma factor that is in prokaryotes. The sigma factor will actually find the promoter first, and then it calls in RNA polymerase. Once elongation begins, you can kind of see this here. It fell off the screen, but once elongation begins, RNA polymerase can do that without the sigma factor. So that sigma factor will fall off and then go find another promoter for RNA polymerase to be attracted to. That is something we see in prokaryotes. In eukaryotes, there are tons of different transcription factors. There are general transcription factors that find the promoter and call in RNA polymerase. Again, you don't need to know about all those different factors, but these here are general transcription factors. That's what they're referred to as. We're going to look at that in a few more minutes when we talk about gene control. A big part of eukaryotic transcription, translation, all the way through, is this processing of mRNA. This is a big difference in eukaryotes versus prokaryotes because prokaryotes do not do this part. There are three parts to it. One is, you notice in our gene area here, we have exons and introns. Introns are transcribed, but eventually we take them out. So the green intronic areas here are removed. Bacterial cells do not have introns, so therefore they don't need to remove introns. This is going to become extremely important when we talk about biotechnology in a couple of weeks. So RNA processing involves three things, and this is all in eukaryotes. It is removing the intron, so you can see the intron is here, but I've taken it out. It is also adding a 5' cap and adding a poly A tail. These all happen in eukaryotes. Now, let's move on to translation. And in translation, for bacterial cells, there is an area on the 5' end which is called the ribosomal binding site. You might hear it called the Shine-Dalgarno sequence. And this is where the ribosome is going to hook onto the mRNA so that translation can begin. for eukaryotes, we don't have a ribosomal binding sequence, but the 5' cap that I mentioned before. So here's the 5' end. We add this huge cap. That is what helps the RNA to mRNA to attach to the ribosome. And then the poly A tail, which I mentioned before, we only see that in eukaryotes. Now we are able, I'm going to unhide a slide. Oh, I can show it to you here. Okay. One of the things that we've taken advantage of, which is also based on a difference between prokaryotes and eukaryotes, is that they both use ribosomes, but what the ribosomes are made up of are different types of proteins, different numbers of proteins, different types of RNA. And so when we create antibiotics, we can create them so that they cripple the bacterial components that build ribosomes, but not the eukaryotic ones. Thank you very much.